So what is mean immobilized enzyme? But basically in terms of immobilized enzyme, what we're going to do here is we want to trap, okay, we want to trap or bind these enzyme onto a fixed structure, for example, alginate. So what we're going to do here is we can make a bit like this. Can you see that, like your boba, like yogurt ball like this? Okay. So what we do, actually, we're going to trap this enzyme in the bit. Okay, so we need to know the reaction, because in the question may ask you how to do this. So you need to know step by step. Okay, so in our syllabus, we only focus in alginate, but okay, don't always think that, oh, only one way to actually immobilize our enzyme. We can use affinity tag binding, we can do the adsorptions on the glass, we can use a cross linkage, we can form the covalent bond. Okay, so that we'll be able, for final aim for this actually is to make sure that our enzyme will bind to a particular solid surface or a particular solid structure, so that we can reuse the enzyme. Then at the end of the process, our enzyme won't contaminate our products. Okay, okay, sorry. Have you guys actually know one of the case, okay, or the disease called lactose intolerance? Basically, a person with lactose intolerance, they might not have the functional lactase. Okay, so if you look at a person, if a person that without lactase, what will happen? We do know that, okay, in our milk, so milk contains lactose. So in the small intestine, we have a lactase. So lactase is going to convert or hydrolyze, again, the lactase, going to hydrolyze lactose into the alpha glucose and beta galactose. We learn about this, okay? Now, why we want to convert this? Okay, why we want to convert into the glucose and galactose? Because they are monosaccharides, they are simpler sugar. And of this, therefore, we will be able to absorb. Okay, we'll be able to absorb this simple sugar into our cells, into our blood. Okay, now, what will happen here is, if a person without lactase, so this person, okay, we have what we call a lactose intolerant. Are you clear? Lactose intolerant basically means that they cannot drink the milk that contain lactose. Now, what will happen to if this person, okay, this individual drink the milk that contain lactose? Now, what will happen here is, first, without lactase, lactose will accumulate. Okay, now lactose accumulation, what will happen? When it accumulates, so means that because lactose, they are water soluble, means that they are polar or hydrophilic. So therefore, they are going to lower down the water potential in the lumen of the intestine. Okay, so we do know that in the large intestine, what will happen? Okay, in the large intestine, okay, so one of the function of large intestine is to absorb, okay, water. But the thing here is, try to imagine this is the cell, okay, epithelial cells, okay, so this is the epithelial cells of the large intestine. This is a lumen. Now, because of the lactose present, so lactose lower down the water potential, okay, and then in the cells will have higher water potential. So now you can see the water molecule actually move out by the osmosis down the water potential gradients. And you try to imagine accumulations of the liquids inside the large intestine lumen, it will cause the diarrhea. Are you clear? It will cause the diarrhea to a person who is lactose intolerant. So it means that if you know your friends having these lactose intolerance issues, don't give them any milk. You have to give them the milk which is lactose-free milk. So this kind of person, they can only drink lactose-free milk. So means that the milk actually without lactose. Okay, so means that we have to help this person to convert or hydrolyze the lactose first. So means that in the industrial process, in order to produce this lactose-free milk, we need to hydrolyze the lactose first. So means that inside the milk won't have lactose, but they have the glucose together with the galactose. So glucose and galactose, no problem, because they can be absorbed in our body or anyone's body, because we do have the transporter or carrier protein to take this glucose and galactose into the cells. So that's why now today our focus here, how we produce lactose-free milk. Okay, so very simple, because the person, lactose intolerant persons, they cannot absorb lactose, they cannot hydrolyze lactose, so they don't have a lactase. We can use a lactase, right? So in the industrial process, logical thinking, theoretically, we can prepare a beaker, okay, a container of milk. So this milk actually contains lactose, theoretically. Then we add what? We add the lactase. Cannot, okay? You got lactase inside. So what does lactase do? Lactase is going to hydrolyze the lactose, okay, into the galactose and glucose. But we do have a problem here. What is the issue if we do this? What are the issues? So first, your final product, okay, your final products, in this case, okay, the glucose, the milk, okay, glucose and galactose now, it's not pure, right? It's contaminated. Why is it contaminated? Contaminated with that? Contaminated with the lactase. Correct or not? Right, lactase is there. So if you want to remove lactase, then we need to carry out purification process onto our product. So if okay, so we have to do the purification onto the products, and this incurred cost. Can I see that? Okay. Now, second, if you say that never mind lah, we don't want to actually, okay, we don't want to remove it. Can or not? Can, but you have to make sure that now the lactase, because normally we don't use a human enzyme. Human enzyme expensive, right? We're going to get lactase. So lactase is from other animal source or other species. So it means that this lactase, you say that never mind, we put it in, okay, we don't want to purify, but this lactase may cause allergic reaction. Because it's a protein from other species. We do not know. It may cause the allergic reactions to the person who drink this milk. Okay, and you may say that never mind, if let's say lactase is safe, no problem. But the thing is, we cannot reuse the lactase. Can you see that? So these three issues now, we have these three problems. First, the product is contaminated with the lactase, right? The lactase is inside the milk. So we want to remove it, can spend more money for the purification. Okay. Second, you say that, okay, now never mind, lactase is very cheap. Okay, we can afford. Okay. Then the lactase may come from other species, may come from a bacterial, they may come from the yeast, they may come from the fungus, they may come from the plant. So this may cause the allergic reaction to the person who drink it. Okay, okay lah, never mind. Lactase actually will come from the very safe. Okay, but the thing here, we cannot reuse. So this problem now, you can see that you cannot reuse. So every time we have to use a new batch of lactase, definitely again cost more money in the industrial process. So in order to overcome these issues, what we need to do? Okay, so one of the methods or one of the way we can actually immobilize the enzyme. So we produce immobilized enzyme. So means that we're going to make sure the enzyme now is on. Okay, enzyme is on a surface. Okay, it's on a surface or a solid. So therefore, okay, if you manage to stick them on the surface, then what will happen here is you will be able to retrieve them, okay, at the end of the process. So what we can do, okay, look at this. First, one of the way we can do it, actually we trap them using the alginate bit. So what we do, so we can prepare one container, one beaker of the enzyme. So in this case, we prepare lactase. Okay, and then we prepare another beaker, okay, or another container. This container contains the sodium alginate. So sodium alginate is aqueous, means that it's a solutions. Lactase is aqueous, also solutions. Okay, so what we're going to do here is we take equal volume and mix them together. Are you clear? For example, okay, like in lab scale, we use 5 ml and 5 ml. So means that we're going to produce a mixture. Are you clear? Mixture of lactase and sodium alginate. Okay, so in this beaker, we have the mixture now. Okay, so now what we're going to do here is, okay, now look at this. We're going to take a syringe, use the syringe to actually, okay, take the sodium alginate mixture here. Okay, we're going to have the mixture. Okay, so contain a mixture. Then we prepare a beaker. So this beaker contains the calcium chloride solution. Okay, so what we're going to do here is very simple. Slowly, slowly. So you press on the plunger here, allow drop by drops of the mixture now enter into these calcium chloride solutions. Okay, so drop by drops using the syringe. Okay, drop by drops enter into this calcium chloride solution. So what will happen here is, now look at this. Sodium alginate, okay, they are soluble, so they are aqueous. Okay, now sodium alginate going to react with the calcium chloride solutions and form what? And form the calcium alginate. Calcium alginate is a solid. Are you clear? Calcium alginate is a solid, and you get the sodium chloride solution. Are you clear? So ion exchange now. Alginate now means combined with the calcium, sodium combined with the chloride. So this calcium alginate is now solid. Now, when it's solid, try to imagine, when you drop this, when you drop the droplets into the calcium chloride solutions, what will happen here is inside we do have the lactase. Some lactase actually on the surface, but they are trapped. Why they are trapped? Because solidification takes place from the outside. Okay, it becomes solid, right? It becomes precipitate because in this case, how it forms? It forms start from the outside. So actually, they're going to trap the enzyme inside the bit. So now we form what we call as the alginate bit. So this alginate bit contains the immobilized enzyme. So my enzyme now, they are trapped. Are you clear? They are trapped inside. Something like you encapsulate, okay, this enzyme. Okay, so what we're going to do with this bit? So few ways we can do it. One of the normal way, okay, we can pack them into a column. Okay, we can pack them into a column. Like for example, you can use a burette. Okay, and then we pack this bit. Pack them nicely. Okay, pack them. So this bit contains the immobilized enzymes, correct or not? Okay, so what we're going to do here is we're going to add the milk that contain lactose. So let this lactose actually runs over the surface of the alginate bits that contains the enzyme. Are you clear? Okay, you read it. So means that this milk will going through, okay. So while they're going through this, what will happen? Reaction take place, correct or not? Okay, so the lactase, the immobilized lactase here, going to break down the lactose into glucose and galactose. Okay, so what we're going to do, very simple, we'll prepare another beaker, and when we open the tap, so means that this milk's going to enter into the beaker, we can collect, and we can do few rounds so that I can manage to complete the reactions. So at the end of the products, now we look at the products. So products definitely free of lactose now. After a few rounds, and we can check, definitely we can check whether got lactose or not. Okay, but what are the advantages in general? What are advantages in general? First, you see that no purifications needed. Why? Because where is the enzyme? Enzymes are still inside the column. Can you see that? It's not mixed with the products. Do you realize it? All right. Okay. Second, enzyme can be reused in this case. We have done the first batch, we can use it for second batch. Enzyme can be reused. So both of these features going to help us to save costs of production. Are you clear? Definitely we can save costs already because no purification, we do not need to spend money to actually purify the products. Second, the enzyme can be reused, so save costs. And the third one, this one, okay, the third one, in this case, generally, immobilization stabilize the enzyme. So means that they can withstand, okay, slightly higher temperature. We increase the temperature slightly, yes, no problem, or slight deviate. Oh, sorry. Slight deviate in the pH. No issues. They can withstand the slight increase in the temperature and slight deviates in the pH. Now, why this thing actually can take place? Why? Okay, because when you trap it, so try to imagine, this is the enzyme under no immobilized, it's not immobilized enzyme. What will happen if you increase the heat energy? You heat it. So means that you're going to break the hydrogen bonds, right? When you break the hydrogen bonds, it tends to denature. Okay? It changes the precise three-dimensional shape. But you try to imagine, you try to imagine now. Okay, now something like this. Try to imagine you yourself. Okay, under very free environment. No one actually we don't use anything to tie you. Okay, and you found out that your hand itchy, right? Bitten by, let's say for example, the ant or mosquito. So you can actually change your shape to actually to scratch on the itchy sides, correct? The side where mosquito bite you, correct? Okay, you have free to move. So therefore, denaturation can take place. Okay, but now you try to imagine I tied you. Okay, because of immobilization. Okay, immobilization. So means that you are tied inside the structure. Okay, something like I use a string to tie your hand, everything. Okay, even mosquito bite you, can you change your shape to move and then scratch on it? You won't be able to do this because you are trapped. So means that the active site actually stabilized by this trapping material. Are you clear? So this is what is the benefits of immobilized enzyme. But again, if you cannot use a too high temperature because if you try imagine that I heat up the solution, what will happen is you vibrate more. right? Kinetic energy is high, they vibrate more. They can actually try to imagine that now you are so itchy already. What will happen? You try to actually move, okay, so that you want to actually remove the spring, and so that you can actually go and scratch on the itchy side. It's correct or not? So in this case, yes, but you must heat up high. Okay, so the question is why we want to increase the temperature. Try to imagine the Q10 effect that we have learned. So any increase of the temperature by 10 degrees Celsius, you're going to double up, okay, the rate of reaction. So in order to increase the rate of reaction, sometimes we won't be able to heat out the solutions too high because you're going to denature the enzyme. But now with the immobilized enzyme, you can actually increase. So for example, without immobilization, you can up to 30 degrees Celsius only. But now, okay, with the immobilization, we will be able to increase to 40 degrees Celsius. Hey, don't look at, oh, only 10 degrees Celsius, but this 10 degrees Celsius allow us to increase the rate of reaction, we double up the rate of the reactions, okay? Because Q10 effects for enzymes, still remember, we talk about Q10 approximately equal to 2. So it means that the rate will be double up for every 10 degrees Celsius of the increase in terms of the temperature, okay? So we look at it, so good. So again, we can actually have no purification needed. Enzyme can be reused, and then immobilizations actually stabilize the enzyme. But what are the disadvantages or limitation? So generally, in limitations of the immobilized enzyme, immobilization enzyme actually decrease the rate of reaction. So why decrease the rate of reaction? So now we have to look at it into two different perspective. So first perspective, if you look at the immobilized enzyme, most of the enzyme, they are trapped inside the beads. Okay, some they are located on the surface, they are trapped on the surface. Can you see that? So means that because of the beads traps the immobilized enzyme inside, so we reduce, can you see that? Only, okay, the only, so how to put this, sorry. So only the enzyme that located on the surface of the beads can react, correct? Okay, only they can carry out catalysis process. Those inside cannot. Are you clear? So only enzyme that located on the surface of the beads will be able to carry out the catalysis reactions. Okay, the rest cannot. Okay, unless my substrate able to diffuse into. Okay, if this is my beads, if the beads actually allow the diffusions of my substrate into the beads, okay, so that the hydrolysis process can take place. But the thing here is when they enter and come out, they take time. So time is actually increased. So therefore, the rate again decrease. Can you see that you need more time for the diffusion to take place? So generally, you can see that the rate of reaction, the limitation here, the rate of reactions compared to the free enzyme, definitely free enzyme, the rate of reactions will be higher, okay, at the same temperature. So in order to overcome this problem, so to overcome this, so what we can do some modification. So what are the modification we can do? So first, okay, we increase the concentrations of the enzyme. How we increase? Before mixing with sodium alginate, so that we can trap more enzymes. Are you clear? We can trap more enzyme on the surface. Are you clear? It will double up the concentration, so it means that it's the same surface area, we are going to have more enzyme onto the surface. Okay, number two, we want to make the beads have small or short, a small diameter, or means that small beads. Why we want to make it small beads? When it's small beads, so means that surface area to volume ratio is large. So therefore, also the diffusion distance will be smaller. What happened to me today? Okay, the diffusion distance will be shorter. Can you see that? So we make the bits smaller, and then we make them to have a high surface area to volume ratio. Therefore, the diffusion distance also shorter. We can increase the rate, even though they need to diffuse, but very short distance only for them to diffuse. Okay, and number 3, because we want to add, because the number of bits correlates with the number of enzyme. So it means that by increasing the number of bits, we will be able to increase the number of enzyme. Correct or not? Try to imagine if I add 10 bits, each bit only have 10 enzymes. So it means that if I add 10 bits and add 20 bits, definitely it will increase the rates of reaction, because we do know that immobilization generally decrease the rates of reactions. Okay? Are you guys clear about this? Okay, so now look at this. So these diagrams, okay, the diagram just summarize what we have talked about it just now. So you can see that we have the mixture of the sodium alginate, okay, and contains the lactase inside. So we dropped the solutions bit by bit, okay, drop by drop into a beaker that contains calcium chloride solutions. Therefore, they form the bit. So this bit actually traps the enzyme inside. Then we can pack this immobilized enzyme or lactase, and then we pour the milk. We can collect. Okay, we can do a few rounds so that we will be able to get a product without the lactose. Okay. So what you need to do now, complete these activities. Okay, I'll give you guys 2 minutes to complete these activities now. So I think you guys have done. Okay, let us look at the answer here. Okay. So first, what we're going to do here is we mix, okay, the lactase, okay, with the sodium alginate solutions. Okay, first thing, we mix the lactase with sodium alginate solution. So U. So after that, we're going to mix. I take this mixture and then add to the calcium chloride solutions, okay, and let the reactions take place, allow us to form the bit. So this bit actually contains the immobilized enzymes or immobilized lactase. Then we can pack this into a column, so T, and then in this column we can actually pour the or add the milk, okay, and let this lactase on the surface of the bits converts the substrate into the products. Okay, we let the solution contain the substrate again, the milk or the lactose are allowed to run over, okay, the surface of the beads, and then the lactase we're going to convert. Okay, so answer UPQSTRV. Okay, so as we discussed just now, what are the advantages, okay, and disadvantage of immobilized enzymes? So first, you can see that the enzyme adhere to the supports, therefore no contaminations to the end product, so therefore do not require purifications. Furthermore, the immobilized enzyme can be reused, therefore the cost of operation also cheaper, okay, of cost of productions. And generally, immobilization help to stabilize the enzyme, can withstand the denaturation of the enzyme when we have a slight increase in the temperature and also slight deviations of the pH, okay? So what are the disadvantages? Okay, one of them, actually, the rate of reactions may be lower. It means decrease in the rate of reaction compared to the free enzymes. Okay, so but we can overcome it. How we overcome? So 3 ways we can overcome. So first, we can actually increase the enzyme concentrations, okay, before we mix it with the sodium alginate. Right? For example, initially I used 1 gram. So now per ml. So now I use 2 grams per ml. Okay, so means that we increase the concentration of the enzyme. Second, we can actually make the bit as small as possible so that they can have the higher surface area to volume ratio. So more exposure, shorter diffusion distance, therefore higher rate. Okay, and third way, we can actually pack more. Are you clear? We don't make them smaller only, but we pack more. For example, okay, if there's a big bit, we use, hundreds of them. Now we increase 200, 300 small bits. Definitely, we will have higher rate of reactions. Okay, so where we apply this immobilized enzyme, we can apply it in the medicines. We can apply it in the food technology. Just now I said that, or industrial process where we need to use enzymes. Some enzymes are very expensive. Therefore, we can use this kind of immobilized enzyme to make sure that we can reuse back the enzyme. Okay, so with this, I have done for the enzymes topics. Okay,